Squid's trying to help.
Moment of truth.
What was this one number-wise?
That would be 55.
Look into it. Look into the thing.
Maybe, can we turn the suction up higher?
I could.
Turn it off for a second.
Turn it off?
Yeah.
It is off.
And turn it back on at 35.
Oh,
There
it
hey,
is.
got
Okay,
it.
now you can turn them off and go back to flush.
All right.
That is one thing I miss in not being on the ship, is being able to see all those extra camera
Yeah.
angles, to see the Niskins fire and the slurp sampler chambers clear and fill, and
Starboard
the right side bio boxes. Yeah.
Well, if there's ever something you want to see, we can bother video by asking to put it on channel 3 while,
Oh no,
while they're
that's
zooming
okay. I just
Can zoom out a
I
bit
just love
if you want, Marley.
seeing all of the chambers become filled.
That's good.
Yeah, we're all staring at it over here, and
Back
you
in
guys
a bit.
don't know what's happening until we say it.
I know. I do know what's happening.
good,
I
thank
just
you.
can't see it.
While we are hanging here, can we grab a Niskin?
Put that, yeah, sure.
All right.
Put the camera back on the starboard wing
Nice
cam.
sponges hanging off that wall.
You want to be set down for a Niskin or hovering?
It can be up in the water column. That's just fine.
Roger.
Does this look like another Pacific Ocean perch down there? Kind of hard to tell.
I think that might be another dark blotch or red band, because I'm not seeing that big lobe that they've got on the lower jaw, but it's hard to tell from this altitude.
So this will be our first Niskin, Niskin 1.
Watch out for that magnum arm. Ready?
Watching. Triggered.
Sweet.
So what happens is there's an elastic cord that will shut the top and bottom of a bottle that's mounted on the side of the vehicle, and that'll capture a water sample from this exact location, this depth, and that water sample will end up in Meredith's labs, and Meredith can explain what she does next.
So I take the filters from the water that are going to be filtered aboard the ship, and I extract DNA from them. So I'm essentially getting DNA from any cellular material that's floating around in the water right here. So from these sponges, from the squid, from this fish we've got sitting down here.
That was 56 running, by the way. I don't
all
know
of
if
these.
you caught. Yeah.
And then I will sequence them in the lab, and I'll be able to tell kind of everything that's within about, approximately 50 to 100 meters of where we are right now. Sometimes a little closer, sometimes a little further, depends on the current. But I'll get an idea of what all the fish are. And nicely, we can also see anything we're missing. So if there's a coral that's just kind of tucked on the ledge system down, we might be able to pick that up. And so it's a good way of confirming what we're seeing in the video, but then also getting a picture of anything we might be missing in the video.
How old is that fish? 200 years?
That's a good question. We're still working to identify it, but you're right that these rockfish can live very, very long lives. We know that at least 140 years for some of them, so quite a bit longer than humans.
One time I convinced Susan Murley that we can age them by finding the tiny harpoons in the side from the 1850s.
For quite some time, their life history wasn't well understood, and they were fished very heavily, and many of the rockfish populations crashed along the west coast. But as fisheries management has become more astute, that many of the rockfish populations have been rebuilt over the past couple of decades. Erica thinks this is either a rough-eye or short raker rockfish, and they can live approximately 125 to 140 years old.
Wow.
Okay.
Thank you for the nice zoom on this guy, the girl.
Like he's got his own patio here on the cliff.
Yeah.
That crab is gonna fall.
Wonder how long he's been hanging out on that patio.
Pretty sweet spot.
Yep.
I'm intrigued by what lies just to the left. Looks like we're on another pretty steep slope here where the crab's headed.
Yeah.
Now, what is the nearest waypoint letter, please?
Sure. It will be D, as in dog. We're just
Thank you.
just like 3/4 of the way between C and D. Maybe 2/3 of the way between C and D.
We may actually want to pick up our pace a little bit,
Okay.
just to make sure there's plenty of time for the photo mosaic
Roger. Yeah.
once we hit the top of this feature.
Yeah, we can definitely pick up the pace and then. Let's see.
Oh, you can see the cutout in the sonar there.
Right. Yeah. I think if I go, well, you're fine. You're directly under.
All right, I'm back.
Roger.
Kind of what? Sweet.
It's a really cool little feature.
Oh yeah.
The nooks and crannies.
So D from here is from Hercules is About 55 meters at 138.
The one there on the nav screen, that's D? Yeah,
Yep.
yeah. Roger.
Nice Swiftia right there that just went over, perched right on the edge. We'll pick that. So that'll be a coral that we haven't seen before that we'll pick up in that DNA sample.
My debris chasing me up the hill there. Oh.
Upward draft.
Oh.
Well, this is just the type of habitat we were targeting. So I'm glad we found what we were looking for. It's just what we were wanting to explore, these steep habitats in the steppe zone.
Yeah, this is awesome. So it's near waypoint L, correct, that we were planning to look around a little bit and see if there was some photomosaic potential.
Are we moving? Yeah.
Oh, would you like to?
Yes, please.
Yep. Bridge nav. Four 0 meters, 140.
Bless you.
Gesundheit.
Thank you.
You need to change your mask now?
No, I sneezed perfectly, so I didn't need to do that.
Looked like it
Excellently
hurt.
executed.
Yeah, it takes a lot of
Another
effort,
big area there.
and now my back hurts.
I did that the other day and I couldn't get my mask off fast enough. Disgusting.
This is a pretty patch.
Yeah, this is a
Some
nice
Phorea,
bit here.
pom-pom anemone, other anemone.
That
Mhm.
yellow
The yellow sponge
thing
out
on
to
the
the right.
far right.
Yeah,
Yeah.
I
there it
missed
is.
it.
Oh yeah, the tiny little yellow sponge
Oh, yeah.
with that thing.
Bright yellow.
Ah,
Oh man.
sorry.
It's too tiny.
You got a little too close.
We'll see another one.
Bridge nav. Is that a move called in?
I see that. How's that possible? Coming up.
Thank you.
You want to look at it? You stop the ship, ready?
Bridge nav.
Can you pan down?
Oh yeah, that's a
Pan right down.
hold
Pan
position.
down somewhere.
That's a daisy wuzzle.
Uh-oh.
See if we can get it out.
Looks like the daisy chain is suffering there.
Yeah, it might be able to come out.
Times like these, I wish Argus can tumble.
Good eye,
out.
Michael.
It's my jorb.
Let's
Yeah.
come up nice and easy.
Yeah, I saw it in down cam, and I couldn't tell if it was that black.
Yeah, you can see a bit of looseness on Argus butt as well, but It appeared like it was.
Sorry, kids on the shore. We're gonna do some tether management here.
What? How much am I coming up?
Yeah, come up easy.
Copy.
Got some clear water here.
The first rule of ROV diving is keep the ROV safe. So
Okay, hold what you got there.
Holding.
There it is in the down cam. So what do I got? Ooh. Ooh.
Ooh.
Mm.
Oh, no, stop it.
It looks like. Oh, it's coming out.
It looks like it's either like, I don't know if it's a proper wrap or if it's like the daisy chain got loose, so it's like doing this weird thing.
Well, the daisy chain's definitely loose.
Yeah.
Is that a turn to port? Can't quite see.
That's a.
Mm.
See you right there, yeah?
Yeah. Let's put some slack in it there.
For those of you watching from home, if you switch to the quad view, you'll be able to see the view from Argus looking down at Hercules. And you'll see the tether that connects those two vehicles.
It's gonna take us a few minutes, kids. It's got time for a coffee break.
All right, I'll be back.
Are you
It's
gonna?
you.
Are you intending on doing like a butt to butt to see if you can stretch it, or?
I was just gonna try and come around and see if it comes out.
Yeah, like a full. Yeah.
Yeah, that daisy's pretty loose.
Should hopefully take it out there.
Would you like me to
What's that?
Slowly come about.
Oh, come up and see if we can see it in the tail cams there.
Yeah. You can see the droop there in Herc butt.
Yeah.
I think it's still there. It's hard to tell. That it, I think it's. You see that? Looks like the daisy or something, or is that the?
Yeah, it's just got the daisy is a bit taut on that bottom part of the loop.
It looks like it's out there.
It's mostly out. It's just the bottom part of the loop there. It's not
Alright.
quite on that.
That's,
There's a lot flopping around there.
Yeah.
oh, it's still caught behind us. See it in Argus cam there.
Yeah.
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 Let's see.
Mm.
Should have taken it out, hopefully.
That might have, yeah. Drive out to recovery. Mode looks just waiting to see it in Argus butt.
Yeah, I'll come up with it again. Stretch it out a little bit.
Yeah, Roger.
I think it's out.
Wish I could change this contrast. It looks like it may be out. It's coming up. I see you.
Just out of view.
Yeah, that's because it was on the
Yeah, I think we're good.
between. It was between me and the first football.
Yes, so it's definitely out.
Is that
Yay.
Still a bit loose on the Argus end, but gonna have to live with that flopping around.
Yeah. Those tend to come loose there
Yeah.
once in a while.
see?
Oh yeah, yeah. It's got a,
Oh, yeah.
but it looks clear. You just maybe need
I
to
know. I don't think
be
so.
taut, and it will. The daisy will pair back with the tether.
Yeah.
But the loop is out, which is good.
Tautening now.
Tautening. Tauten.
Tauten.
I don't think.
All right, it's tight and the lights didn't go out, so it's
Yeah.
encouraging.
I don't know.
All right, where were we?
We were...
Somewhere around here.
Somewhere on a cliff.
Yeah.
We're going down.
No, hold what you got there. Let me go find the... Let me get back over to the cliff there. Okay, I think we're all good. We're just headed back to where we left off.
Great. Sounds like we are headed towards site D, waypoint D or E.
Roger.
That's right, we were headed south, weren't we?
Southeast.
Where did I leave off? I left off there.
I think that was the drive out. Yeah, you were somewhere in here. Give you a view of it there.
come over and get the cliff so we have something to look at, and then fly back to the.
Roger.
Go back around the way we came.
There it is. I see it.
Come down 5 for us, Michael.
Own it.
Is that the old boy again there? Maybe.
Yeah, yeah, it kind of looks like a pattern. We've zoomed in
Oh.
on that boulder before.
I recognize you all.
Oh, deja vu.
Been here, seen it.
We know
done it.
where we are now.
Sorry, guys. I came way to the north. See if I can take a different pattern.
Asking the pom-pom to act as a marker for us.
Mm.
Keep coming up, Mike. Come around the.
There's our little Swiftia.
Mhm.
And Jenny, if you're on the chat, Nicole has a question.
Those pom-poms are just so ginormous.
You're making some good time now.
Yeah.
Yep, cruising.
Cool stuff to see cruising along here.
Recognize all that.
Now this is pod racing.
Oh,
Okay, you got me again?
City.
Okay, so roughly back where we were.
Yeah.
Kind of, sort of. Different depth, probably.
Ready for 145? Move.
Roger.
So many.
Pom-pom
Great job.
heaven.
Yeah.
40 meters, 145. Thank you.
Like, why here, you know?
You're tugging.
Everything was just right.
Yeah.
Whoa, look at all these.
Just tuning in, Mike?
Yeah.
Apparently so.
Hey, I'm on Tether Watch.
True.
That's right, yeah. And
We appreciate you.
good tether watchman you are.
That's the second time he's saved the show.
Yeah.
I appreciate me too.
Good.
Thank you, Tetherman.
That's your new superhero name. Tether man. Can't wait to design the costume.
He knows how much work it is to change that thing out. So.
Yeah.
And build a new one.
It
It's done both.
will involve daisy chains,
Yeah.
the costume.
Oh yeah, absolutely.
Get some of those yellow
Those
football
of you at
floats.
home,
The insignia could be like a pretzel undoing itself.
Yeah. Okay, yeah, just wait. Ship's moving.
Waiting on a ship move, you said?
Ship's moving.
Okay.
145.
what is that yellow sponge
Yeah.
in the center?
Katie said
It's
maybe
looking more at
egg,
the, is
gate
that a
egg
shark
case.
egg or a,
Yeah, it does look like an
They're
egg
There's a
case.
gate egg
different.
case right on the right-hand side of the
It
screen.
is, yeah, good.
yeah, there's an egg case
Yeah,
there.
there are a couple.
Yeah, looks like one a
There's
bit
another
farther
one.
too.
We've got a
Yeah.
Swiftia down on the bottom left. It's got its gate eggs on it.
Yeah.
Oh, yeah. Could we zoom on the yellow sponge right by the lasers, please?
Sure. Go ahead for laser zoom there, Marley.
Ooh. Pretty.
It's pretty.
That's one we haven't really seen yet, I don't think.
Come down 5, please, Mike.
Copy.
Abby doesn't
Abby
recognize
doesn't recognize
What?
it. That means we should probably collect it.
Okay.
Okay, you can zoom
We
out.
do
I'm
have
gonna
unknown
back away
species
till
on
we
our
get
list.
Okay.
Yeah.
I'm gonna back away till we get the boat a little closer there. I'm struggling to.
Yeah, because when we're operating in these
I'm gonna
deep
hold and
areas,
move 90.
we can
Bridge
and
nav.
Roger.
Abby is noting
Come down
something
another
about
5, Mike.
permit,
Another
Jenny,
5.
on chat.
Yes, please.
Hold position, and then we will move 20 meters bearing 070.
You are.
Yes, in general, with the permit, we don't like to collect the first or the only,
Yeah.
but for species that Abby doesn't recognize that could be new to science, that is a bit of a trump card.
yeah, with some of the deep sea species,
you're
because
gonna
they're,
reset.
so little work gets done in these areas, we do
There we go.
try to put in our permits to collect unknowns so that we can learn what they are and add them to our voucher collections. Both for genetic and taxonomic ID.
I'm all paranoid now.
Am I gonna have
Looks like
enough
I have
to
the big
park at the
hospital on
single?
left.
And Abby would like
I'm
another
going
zoom when
that
we get
way.
the chance, when we're in a good
Coming
position.
towards me?
Yeah-ish to the right, but yeah, 070. Argus
Yeah.
is facing 045-ish.
Move
So
closer to you.
I'll be closer.
I'm gonna park on this heading here. Maybe on the little shelf here would be nice.
Yeah.
And I'm starting to move.
Zoom in there if you want, Marley. Should be good for full zoom there, hopefully.
This is a good angle.
Osculum.
Yeah, we are like on just on
Look
top
at
of
you
Beautiful.
with that bio knowledge, Dan.
That's good. Good on the zoom. Maybe out just there. Perfect.
Awesome.
I'll start. Are we gonna slurp?
Um,
Or full like bio?
I don't know if that's a grab. Let me see what Abby says. Jenny, you have any input on that?
Sticky finger grab.
Yeah,
I'm gonna
could-
defer to Abby, but
could-
this.
Do the sticky fingers open wide enough to try that?
Yeah, you gotta kind of force it open around it. I don't know how fragile it is.
Yeah, I guess none of us do. Abby's thinking more likely some kind of grab. Do we think we could try to very gently grab it?
Yeah, I'd
Alright.
just grab low and get some
Sure,
of the clay with it and
let's
get his
do that
kind of
then.
his what do you call it? His I want to say his root, but that's not right.
Stolen type
His
thing.
foot.
Okay, yeah, let's do a gentle grab.
Copy. Group 4 straight. You show me the minute, Mike? You can zoom in just a bit there if you want, Marley. Yeah, perfect. Okay, chase the end effector out.
Yeah.
Show me the victim there. Where is it?
There it is.
There we go.
There, it's not so drunken.
Nice.
Such incredible dexterity.
Oh, stop squeezing.
Yeah. Is front easy to get to? I don't want to risk dropping it trying to get to starboard.
Yeah, front's definitely easier.
Okay, let's do that.
Porch out.
Stand by.
Hey, let me know when you're ready.
Let's do forward A. And Rennie, this is sample 057.
057, roger.
Yep, should be good for porch out there. Oh.
Oh no. Sticky.
I don't know what happened to Sticky there. Should have rode, I think his hose.
Very jiggly.
I'm pretty sure we just made Abby's day.
Yay!
Not in the box yet.
Yay.
Yeah.
I have faith, Dan.
It flies away.
Yeah,
somehow.
is it a floater?
Yeah, we'll find out if it's a
It
floater.
just swims
I have not
away.
hit submit yet.
We should be all right.
No,
we
it
got
looks
our,
like
we got
it'll
our
be
nose
good.
in the breeze there.
Yeah,
Sinker.
nice.
What's happening there?
Sweet.
Okay, close. To do some sticky finger maintenance here.
Durr.
Oh, because you didn't have it.
It must not have been all the way on the magnet. Really?
Ooh,
That's not gonna work.
that's a ways to go, Ren.
See what I'm looking at?
Yeah. How far is it to L?
Oh,
650.
watch out for the Alvin.
What was that?
650,
650.
and that's a straight shot.
Yeah, so we'll have to pick up the pace a bit, huh, to get to L.
Yeah.
Probably.
Is that what you're saying, Nav?
If it's still another 5 hours of this dive, yeah. 4 hours, yeah, something like that.
Or and change, yeah. It won't take us long to surface, luckily.
Right.
I think
We'll
it's
have
on.
to get to L and then get up to our
I
target.
think that's good.
Right.
Yeah, we can definitely pick up the pace. I think I was going closer to between 0.1 and 0.2, but not there. I can.
Yeah, we can pick up the pace of the ship. It'll just be if we see a cliff, it won't be, we can stop at it. We have to kind of breeze past it. But in order to get to L, that's quite a bit of distance to cover. Because we then have to get to L and then get up to the top and have time for a photo mosaic. So it's not a lot of time.
Right.
How far away from the photo mosaic spot?
650 meters.
Half a knot, that's half an hour.
I can tell you.
Or an hour.
650. Half a knot, it's an hour 20. If we go there straight. So we don't have to go.
Hour 20. We won't see it on our shift.
Nope.
No.
But that's a straight shot. So we'll see if we can still maybe keep on our targets. It's up to you, science, however you want to go about it.
Yeah, let's just see if we can maintain a fairly steady pace. We'll try not to do too much with sampling or zooming on the way.
Okay.
And
Yeah,
see
can do.
maybe after when we get to the watch change at 4 o'clock, they may take over and drive the bus a little more quickly than,
Okay.
than
okay.
Meredith and I have.
You're doing great.
Oh, I thought you were talking about me. I was going to say we can go faster.
Don't tempt the man.
Oh no, that's all on us. It's just hard when we have a window into this world not to really gawk and gaze and
Oh yeah,
take
absolutely.
time to look.
I'm totally like Ferdinand the Bull. Like, ooh, flowers.
Yeah.
Yeah.
Got more Swiftia there.
This is 0.2 of a knot here, according to our instrumentation. Probably 0.3 now.
Oh, wrong one.
Seems like good steady speed.
Yeah, it's 0.3. It's,
Do you still want to follow all the points on the way there?
I would say follow your instincts. We're not
Okay.
super tied to those particular waypoints, so
Okay.
if you see a vein of interesting visuals, please take advantage of it.
Okay. I think we can go
Ooh,
step
octopus.
south.
We're not zooming, but
Yeah.
we're looking.
30
Oh, what?
meters, 40 meters south.
oh, you sure
He's
you don't want to
sneaking
zoom?
along
Yeah, we got
there.
time.
Cool. That's a cool one.
We barely started.
So
Bridge, nav.
the problem when I move at 0.3, you'll see, but there's I get a kind of a dust cloud
40
following me.
meters, bearing south.
The one in front wasn't from me. He looks angry.
I love the way he's moving. It's so funny.
Just like,
Like crawling like a toddler.
Trying to sneak away.
Yeah.
I go slow, maybe they don't see me. Oh, they've got all the horns up that are like kind of mad.
Getting all tangled up.
Let him crawl out of the screen and then we'll go.
wrong button.
Okay.
I called in a south move, and then we'll be moving southeast after that.
Yeah, we can hit. Definitely hit D there. Follow along the kind of the contour here.
Roger.
Yeah, that's flatfish really blending.
It's more 0.3 there. Survey speed.
Yeah, did you guys want to do another transect at some point, Science?
Actually, moving at this speed, like just like this, would be a great way to do a transect just as we move along. And if we're on transect, we can't stop and sample anything. So
Perfect.
Should we call it that now,
How
or?
convenient.
You can, yeah, whenever.
Can do it, yeah. We're at D, so we can start at D and do a transect if you want.
Great. If it's a good line, let's do it.
I think we go D to G. That might get us there quicker, unless you're really interested in E and F.
No, that's fine.
Okay, we'll do
D to G sounds good.
And D to G is 160 meters, something like that. So after this south move with Argus, I will move the ship approximately 147.
Roger. 147.
When you're in a
Skipping
good
E.
spot to start the transect, let's drop the start.
All right,
Marker.
T2B.
All right, yeah.
You'll have to move him a little faster, Rennie, half a knot probably.
Okay, I'll bump up to half a knot once. So this
Yeah,
moves.
I'm at the end there.
Yeah. Moves nearing completion. What's our?
I'm gonna put bubble cam
What's
back
a
on
bit-
the right hand side there. Thank you.
Like that?
Yeah, right in there somewhere.
Just set you up here, Dan.
for anyone at home who wants to get a tiny taste of what everyone in the control van sees with all their different camera angles, check out the quad view. And they often have a camera up there that has a view into the van where you can see all the different monitors.
And after a dive, there's usually a camera on in the wet lab where you can also watch scientists processing the samples that come up.
Yep.
Fun thing to do too.
I think when we're processing cores, we're just out of view of the aft cam. But today we'll be more in the lab.
Oh, we can move it to watch you process cores.
Not a request on my part, but
Lila, can you
It's a muddy
describe how you
job, but
process the
somebody's got to
cores?
do it.
yeah, sure. So we're kind of splitting them up into 4 different types. There's, well, sometimes we can use the same core for multiple different things. The 4 different things that we're getting
Come
out
down
of
a
the
bit, Mike.
cores are rare
Oh,
earth
you already are.
elements, the B&B, which are kind of
147, roger.
kind of the more pharmaceutical applications, looking for new natural products. And then there are also some cores we use to look at macrofauna. We used to look at macrofauna, and then the 4th kind of thing we're looking for is microbes. So it's a lot of just slicing, like 1 centimeter slicing, and doing various things with that.
Awesome, thank you. And were you able to process the bonus seep push cores? I think there were 3 of them, right?
From this last dive just now? Or, the one from...
From the Biogenic 2 site? The seep that we happened to
Down
cross.
The
5,
one that you
Mike.
happened to cross?
Down
Yeah.
5, please.
Yeah.
Yeah, those they're processed. We're not really going to get to take a closer look at them or really process them until we're back in the lab, or at OSU. But we did section all those cores, and they're currently sitting in various freezers. For one of them, I think we sieved macrofauna, and that's sitting in the fridge. So some of the B&B, or maybe it was the rare earth, some of them are in vials preserved with ethanol. So they're all sitting in various containers now, waiting to be analyzed further when we get back.
Awesome. Very exciting.
Yep.
Thank you for describing it.
Sure.
I also have some good action shots of that process, some good photos of you guys doing it the other night.
Cool.
The guy ran out of leash there.
Yeah, we're getting close over on the.
Yeah.
Come upslope a bit.
I'll come, sorry guys. I'll come to the left and come up the hill a bit. There must be a valley here somewhere, right? My left should be uphill, but it seems to be downhill. What's going on there?
Your left should be downhill.
Yeah, but that's Argus's left too, right?
No.
Huh?
Yes.
Is that Argus's?
Argus is right. And
How can that
Argus's left is downhill and Herc left. We're both facing the same direction.
Yeah.
So all downhill, all uphill. Downhill left, right, uphill right. That makes
Right,
sense.
so if we want to put, I lost the plot there. I want to come uphill if you want to get Argus further away. Is that right?
That's correct.
All right.
And now the ship is moving forward.
That whole left and right thing just kills me.
All right.
My wife and I argue about it all the time. So, you know. Yeah, so I come to the right to come uphill.
Yep.
That doesn't jive with our course over ground too well, but let's see what happens.
Yeah, and we
Folks
can
to the
probably all
southwest have a
move along there at that.
What's that?
Here, let me
So the
fix
folks
this
at Southwest have some methods for just dealing with what happens if you get pulled off the wall or run out of tether or anything. They usually just add a few seconds or just, adjust where they're doing the transect on a little bit.
Okay, we should be
Try to
good
make
there
sure.
to.
Yep.
Happy with that? 20 meters away from the wall.
Yep. You're just
Okay, cooking with gas at 145.
Roger.
Been passing a lot of fish, eel pouts, lots of rockfish, flatfish.
So you actually do it. You might slow them down a little. Usually ask for half and you get 0.35.
You're like dragging in the dirt.
Thank you. So I have that camera there.
Squishy fingers. Where are you?
Do you want me to slow down then, Dan?
Yeah, I think so.
He's
He's actually doing 0.5. Last night it was 0.35.
Let's continue on this same bearing 147, but slow speed to 0.3. Thank you.
It's a very camouflaged flatfish there.
Yeah.
That, I guess we're going uphill, so I can.
Yeah.
Ooh,
Another stony head.
I got a big wrap on 6.8.
What?
What's that?
I got a 6.8 wrap.
How did that happen?
Yeah, when did that happen?
Yeah. I have to come about.
Not believing, unless you came around the other way.
Possibly when we
Must have.
reacquired, yeah.
Yeah. No, we were all sorted after that.
Yeah, right after that, but then we sampled right after and we
Oh.
swung
And then we did
around.
a
Yeah,
thing,
that
yeah.
could have been it.
Not so worried about the wrapping the down wire, so we are the tether.
So if you kind of ease towards the starboard, that'll get us more in line under.
I can
Yeah,
I can come up, take it out, and come back down.
thought I wanted to go to the port to come up the hill.
Nope, starboard is up the hill to the right.
So I want to come uphill more.
Yeah.
Roger.
Well, there's what's kicking up all our dirt.
Yeah, that's good.
What?
Keep that track.
I was amazed at how much fine mud will stick to a very steep wall.
It's mucklicious.
A lot of Rathbanaster.
Yep. Must be some good food for them right in here.
I probably exceeded my speed limit for a second there just to get out in front of Argus.
I'm glad to see the sea stars fairly abundant down here. The Pycnopodia, which is the sunflower star that occupies shallower depth zones, has really, really had a major population crash in the last few years. So cause for concern, but clearly the Rathbanasters are still fairly abundant at these deeper depths.
Was that affiliated with any kind of wasting disease at all, or?
That's what
That's from the waste-in disease.
people think, yes. There's actually a study going on
They were
right
one of
now.
the species that was hit earliest.
Did you just flip around there, Michael?
Yes, I am. I couldn't keep him in line.
So now you're tail to tail?
I'm coming about tail to tail, and now I'm coming back around.
Oh, you're doing the flip. Understood. Yeah. That was swift.
Boom, like it never happened.
Let me know if you're going to take a turnout, because
All right, we've covered about 120 meters.
Hey, thanks for the check.
Yep.
just so I know to keep the delta tight or keep the space out.
Copy.
If we do it too close, we'll wind up with a hockle.
Ooh.
Wow.
What are those?
That's a line.
Oh, really?
That's fishing line.
line.
Look out.
Yeah.
Fishing line with a lot
Lots
of
of life
Perea
on that
going on
line,
it.
though.
Fishing
That's
line.
fishing line that's been there for a good long while.
Yeah.
Looks like it, yeah.
Oh, yeah.
Still see the harpoons from the 1800s.
Nice. Call back.